Hello, my name is Charles Stock, and I'm a research oceanographer at NOAA's Geophysical Fluid Dynamics Laboratory, where my work involves the development of Earth system models and the application of climate and Earth system models to problems in living marine resource science and management. As the second of these topics I would like to focus on in my talk today. Specifically, I'd like to talk about potential applications of Biogeochemical Argo to living marine resource science and management decisions across time horizons. I'd like to thank a number of colleagues, including Barbara Mulling, John Dunn, and Jorge Sarmiento, for their comments on this talk, and also conversations over the course of several years that have shaped my thinking on this topic. I'd also like to thank the organizers of the Global Biogeochemical Argo Fleet Knowledge to Action Workshop for the opportunity to share some thoughts with you today. And lastly, thank all of you who are listening in for your time and attention. I'd like to begin by emphasizing that environmentally informed decisions across management time horizons are needed to sustain living marine resources in the coastal communities that depend on them. This plot summarizes the range of decisions required in the fisheries management sector. The x-axis corresponds to time horizons from the days to weeks scale that characterize industry operations, out to the multi-decadal to century scales characterizing long-term industry capitalization and rebuilding plans. The y-axis provides a characteristic spatial scale at which these decisions are often made. Living marine resources are strongly impacted by weather and climate phenomena across these time horizons, the number of examples being provided in the cloud bubbles on this plot. My plan for today's talk is to take you through a number of examples of how we're trying to integrate environmental information with these decisions, talk a bit about how BGC Argo might help us improve this process, and also talk a little bit about what aspects of Biogeochemical Argo might limit its utility in this context. Before going into examples, I'd like to underline a point made by several other speakers in our session, and that is that living marine resource management is not just about commercial fisheries. Decisions must balance fisheries goals with a desire to support recreation and tourism, protect endangered and iconic species, maintain a healthy overall ecosystem, and numerous other considerations. It's difficult for any two people to agree on the perfect balance of these considerations, but the odds of achieving an agreed upon balance successfully are greatly enhanced by knowledge of what controls the distribution and productivity of living marine resources. A common theme in the examples that I'll raise in this talk is how environmental information may be able to help simultaneously improve fisheries and conservation outcomes by helping to provide better answers to these critical questions. My first example comes from the West Coast, where a drift gillnet fishery targets broadbill swordfish. Drift gillnets can be over a mile long and often lead to unintended bycatch that can include endangered and protected species. At times, bycatch limits are reached so quickly that swordfish catch ends up falling well below quotas, resulting in a downsizing in the fishery. In response to this challenge, Dr. Elliot Hayes and colleagues at NOAA's Southwest Fisheries Science Center have developed a real-time habitat modeling system called Ecocast. This is composed of a series of habitat models for swordfish and prominent bycatch species, including leatherback turtles, sea lions, and blue sharks. The habitat models are based on statistical relationships between the observed occurrence of each species and satellite information, including things like surface temperatures, chlorophyll, and sea surface height variations that are available in near real time. Ecocast can then calculate areas where fishers are most likely to capture the target species without capturing the bycatch. Outputs are delivered in heat maps like the one you see here on the right-hand side of the slide, where blue areas indicate high catch of the target species with low bycatch. Results of Dr. Hazen's analysis suggest that the information provided in plots like this could enable a reduction of closures by 2 to 10 times without increasing bycatch, providing a nice example of where additional environmental information may be able to simultaneously improve conservation and fisheries outcomes. A limitation of this study, and most others like it, is that they're often forced to use statistical relationships with surface data that's measurable by satellite, when fish, of course, are mechanistically responding to the three-dimensional ocean environment, including both physics and biogeochemistry. There are cases where models based on surface variables can be quite skillful. There can be others where there are meaningful differences that can affect applications like the one I just showed. This figure is from a paper led by Dr. Barb Mulling, which contrasts the habitat distributions predicted by two different habitat models for bluefin tuna in the North Atlantic. The top panel shows a mechanistic habitat model based on metabolic considerations informed by the full temperature and oxygen profiles. And the bottom model is an empirical model based primarily on surface data. The two models actually do quite similar during the contemporary period. The heat maps on these plots are indicative of the favorability of the habitat, where the warmer colors indicate higher favorability. The dots indicate the catch per unit effort in different regions, with larger dots indicating better catch. So ideally, you'll see a nice alignment between the large dots in the figure and the red regions. While the patterns in both models are consistent in many ways, you can also see fairly significant differences that could affect bycatch calculations like the ones we just talked about. BGC Argo, either on its own or integrated with ocean state estimates, had the potential to reduce uncertainties associated with these sorts of applications and move models towards a more mechanistic and reliable basis. However, one of the other things that this figure highlights is that in order for the full effectiveness of BGC Argo to be reached, we also need fisheries measurements at similar scales to the ones that we're creating for the biogeochemical measurements. If we have tremendous physical and biogeochemical measurements and are still stuck with very coarse scale fish measurements, then the value of that added environmental information will be muted. The example I just discussed was essentially a nowcast. It was a near real-time product intended to inform industry operations. Now I'd like to move forward in time and think about how seasonal to interannual predictions are being used in the fisheries arena. Seasonal to interannual predictions are based on the premise that if you have knowledge of the current state of the climate system, particularly the ocean in our case, and you combine that with a skillful model that is capable of predicting how that ocean will evolve in the future, you might be able to anticipate changes on timescales ranging from a few days to a few years. The initial estimate of ocean conditions is derived by assimilating information from the Global Climate Observing System into a global climate model, which is then used to predict forward in time. There are also statistical approaches used in this process, but the result is the same: an estimate of the ocean conditions with anywhere from a few days out to a decade. The models used to do this are often coarse, but even these coarse models have already proven useful for a variety of fisheries applications. The example fisheries application on these seasonal to interannual time horizons that I'd like to share with you is that for the Pacific sardine in California. Small pelagic fish like sardine are notoriously volatile. The Californian Pacific sardine is no exception. It was once one of the largest fisheries in the world and fueled a thriving economy in cities like Monterey. However, a catastrophic collapse in the mid-20th century, which was linked to a combination of climate and fishing factors, devastated that economy. The question now is, can we use global ocean forecasts on seasonal to interannual timescales to help us avoid outcomes like this one? One thing working in our favor is that there is a reasonably robust relationship between sea surface temperature anomalies in California and Pacific sardine recruitment, where recruitment is essentially the number of young fish that are entering the fishery each year. I'll come back to the reasons for this relationship a bit later, but for now I'll just mention that the relationship has proven reliable enough that current and recent SST anomalies, sea surface temperature anomalies, have been incorporated into the catch advice process. With this challenge in mind, Dr. Desiree Tomasi and colleagues asked the question, what if we add information about future conditions into the harvest control rules for the Pacific sardine? She contrasted the catch and the standing stock biomass resulting from harvest control rules that used three different approaches. One included no environmental data, which is shown here under the no SST case, where SST is sea surface temperature. A second adopts the approach that's presently taken, which is to consider the recent past SST environment over the past 3 years to inform the harvest control rules. The third considers the past 2 years of environmental data and then adds predicted conditions for the following year. These two plots show the yield on the left-hand side and the stock biomass on the right-hand side, which can roughly be equated with the fisheries goals on the left and conservation goals on the right. The strategies from bottom to top are no environment, past environment, and past and future environment. And you can see the third case, when it harnesses that skillful prediction of future conditions, attains a higher level of both the yield and the stock biomass on average than the other two cases. Now, this may seem like we're somehow cheating, like we've violated some sort of conservation law here. But the key again is that we've introduced new information, new data, which has allowed us to make better decisions to simultaneously achieve better outcomes on both the yield and stock biomass fronts. The results of the Pacific sardine study give cause for optimism. However, as was the case with the first example, the Pacific sardine case study also had to rely on an emergent relationship between an easily measured physical surface variable and a potentially complicated fisheries response. In reality, SST is likely a proxy for diverse ecosystem changes that cause shifts between sardine and other forage fish species like anchovies. For example, work by Dr. Ryan Rikacheski and Dr. Dave Chuckley suggests that the fluctuations may actually reflect subtle changes in the productivity environment of the California current related to the prevalence of strong coastal-driven upwelling versus more gentle upwelling in offshore waters related to the curl of the wind stress. These are obviously not going to be completely reflected in a simple relationship with SST, but there could certainly be an emergent relationship that could be robust over a period of number of years. However, that relationship could break down in the future, especially with things like climate change, and it's critical that we start to move towards more mechanistic models of fisheries responses. By providing three-dimensional information on critical physical and biogeochemical drivers of fisheries changes, BGC Argo could help us move from emergent correlations to mechanistic predictions. Another area where BGC Argo may prove beneficial to fisheries applications on the seasonal to decadal time horizons is by improving ocean biogeochemical prediction systems. There are now numerous efforts to augment climate prediction systems that previously focused solely on physical properties to include ocean biogeochemical and other phenomena. The process of making a prediction in this case is very similar to the one that I described below for the physical case. The global climate model is simply replaced with a global Earth system model that includes a much broader set of Earth system processes, including ocean biogeochemistry. New elements of the Earth observing system become germane, including BGC Argo. The ocean state estimates that are developed include both physical and biogeochemical properties, as do the resulting predictions that can be used to inform fisheries decisions. We're still in the early days of developing these systems, but progress to date has again revealed some cause for optimism that they may be exceptionally useful in the fisheries context. I'm going to show you some results from a system that has added the ocean biogeochemical part to a physical climate model and carried over the physical assimilation from a previous generation of the model, but is yet to consider the biogeochemical assimilation that could improve the system further. What I'm going to show is that even this system, before including BGC Argo, has provided skillful biogeochemical predictions in many areas that are fisheries relevant, and argue that these could be improved even further with a robust array of BGC Argo measurements. I'd like to start by discussing some results from idealized predictability studies with this system. Predictability studies are conducted by starting multiple predictions that differ only by a modest perturbation to the forecast initialization. These have suggested that biogeochemical signals are likely predictable for very long time horizons, particularly for subsurface quantities, where it can take several years for unpredictable random processes to overwhelm signals rooted in the common ocean initialization. This plot compares the predictability of three different fisheries relevant quantities. The rows are the sea surface temperature in the top, nitrate just below the euphotic zone in the middle, and oxygen just below the euphotic zone on the bottom. Columns correspond to lead times, which are effectively one season ahead on the left panel, 1 year out in the middle, and 2 years out on the right-hand side. The warm colors indicate close tracking between the predictions, which again only differ by a small perturbation to that initial state. If we look at SST, the prediction skill erodes rapidly after the initialization. This reflects the fact that relatively chaotic and unpredictable weather scale variability in the atmosphere starts to obscure the common signals in the SST initialization almost right away. The initial information in the subsurface biogeochemical quantities, in contrast, holds across many months and out to 2 years, suggesting that initializing the ocean state accurately could yield skillful predictions across several years or more. Predictability on these scales would be well aligned with harvest control advice that is often done on those time scales. Idealized predictability studies are useful, but what is really needed for living marine resource applications is the capacity to predict observed fisheries relevant patterns of physical and biogeochemical variation. The first challenge to such efforts is a paucity of biogeochemical time series with which to verify prediction skill. BGC Argo may eventually be one of the keys to resolving this limitation by providing more reliable global picture of biogeochemical fluctuations and trends. In the meantime, satellite-based chlorophyll estimates, which now span over 25 years, provide a good starting point. This plot shows the degree to which chlorophyll anomalies from extensive retrospective forecast experiments compare with observed anomalies over the 25-year record, with warm colors indicating consistent observed and predicted patterns. The top plot summarizes the skill of one season ahead predictions. Areas of skillful prediction are evident in both tropical and extratropical regions. The bottom plot zooms in on each of the box regions in the top panel and organizes the model skill by the month during which the forecast was initialized on the x-axis, and the lead time of the forecast out to 24 months, creating this checkerboard of skill. This took nearly 300 ensemble forecast experiments and over 3,000 individual ensemble members to build this complete picture of the skill over the 25-year record. The key thing to take home from this is that skill extends out to 1 year or more in many of these regions, consistent with the results of the idealized predictability studies. Furthermore, when we dig into the mechanisms that underlie this skill, they're often related to subsurface nutrient signals, anomalies in things like nitrate, phosphate, and oxygen, that are exactly what BGC Argo is capable of measuring. Similarly promising results were found for a number of places where subsurface nutrient oxygen patterns have been measured directly over the course of several decades. Comparison against subsurface oxygen collected as part of the California Cooperative Oceanic Fisheries Investigations Program, or CALCOFI, for example, revealed consistent observed and predicted oxygen declines from 1990 to 2010, though the predictions do suggest a stronger rebound than observed after 2010. Finally, exploration of fisheries patterns suggests that the addition of biogeochemical drivers will present new opportunities for fisheries management applications. This plot, for example, assesses the capacity to predict observed fluctuations in fish catch in the Agulhas Current located on the eastern side of South Africa. Like California, the fisheries catch in this region has experienced prominent variations associated with changes in the distribution and productivity of sardine, with catches peaking in the early 2000s. This increase could not be anticipated by a model that considered just sea surface temperature. It could, however, be skillfully predicted by a model that considered both sea surface temperature and chlorophyll. Thus, the addition of ocean biogeochemical properties has allowed us to move from the use of just emergent SST relationships, like the one in our first example, which is reproduced here in the upper right, to a more comprehensive prediction capable of directly accounting for fluctuations in ocean productivity and other factors depicted by the schematic in the lower right. Examples from seasonal to interannual applications highlight the potential roles for BGC Argo in improving ocean state estimations that can be used to verify model predictions, and potentially improving BGC predictions themselves by helping to initialize high-memory subsurface biogeochemical signals. These examples also highlighted a role of BGC Argo in helping to improve the mechanistic underpinnings of fisheries productivity and habitat models. This last contribution comes to the fore as we look towards climate change. We can't do extensive retrospective forecasts like the ones described in the previous slides to establish the skill for century-scale climate change signals. Instead, confidence has to be built upon the ability of the models to capture past observed fluctuations on decadal timescales, and the construction of models on robust physiological and ecological principles that we expect to hold under climate change. I think BGC Argo can play a prominent role in helping to build both of these pillars. For a quick climate change example, I'll go back to the work of Dr. Barb Mulling on bluefin tuna in the North Atlantic. We noted that a habitat model featuring explicit metabolic dependencies on subsurface properties measured by BGC Argo, and a habitat model adopting a more common statistical approach based primarily on surface data, yielded differences in their estimated distribution in the contemporary ocean. It generally explained observed catches in the contemporary ocean similarly well, was shown here in the left panels. The two models, however, differ significantly in their projected climate change responses, with a biogeochemically driven mechanistic metabolic model anticipating enhanced habitat in many areas of the temperate and subpolar North Atlantic, which is indicated in the plots on the right by the broad swath of yellow in the upper right plot, while the more standard statistical approach had little evidence of this increase. The 3D picture of ocean habitat provided by BGC Argo and the improved understanding of fisheries habitat this could yield may play a critical role in reducing uncertainties like these, subject to the caveat that the improved estimation of fisheries habitat that technologies like BGC Argo can provide needs to be coupled with improved capacity to observe the fish whose habitat we're attempting to understand. We've now completed a quick tour of fisheries decisions across time horizons and discussed some of the ways that BGC Argo might be able to contribute to better decisions on these timescales. Before concluding the presentation, however, I'd like to briefly address one more critical issue. Namely, the majority of fish that we catch are in coastal waters that aren't always well sampled by BGC Argo, as indicated in the plots on this slide. Now, you might be asking whether we wasted our time talking about all those fisheries applications, and the answer is definitely no. Living marine resource management, as I said before, is actually much more than just fisheries. There are also many high-value stocks, such as tuna, that are in offshore waters. Furthermore, many coastal areas like the California Current have extremely narrow shelves, so BGC Argo can operate relatively freely in those systems. Perhaps most critically, coastal waters around the globe are reshaped rapidly by offshore waters that are sampled by BGC Argo. This plot shows the estimated mean time that water parcels have been over shelf areas, defined in this case as areas less than 200 meters deep, for different coastal ecosystems around the world. The highest ages, in red, are a few years, and the timescales for many ecosystems are substantially less. The implication is that any attempt to predict the coastal ocean on timescales comparable or beyond its residence time will require accurate assessments of the anomalies along the boundaries of these systems. Thus, even for areas that may be difficult for BGC Argo to sample directly, it may have an important role to play. As a tangible example, BGC Argo could provide critical information for nascent efforts to develop a nationwide infrastructure for physical and biogeochemical prediction in U.S. coastal waters on seasonal to multi-annual timescales. The grids shown here are prototypes developed for this purpose. Critical questions for whether BGC Argo can be useful for this application include whether the measurements are precise enough to constrain relatively subtle seasonal to interannual anomalies that can nonetheless generate large ecosystem responses, and whether there's added value of BGC Argo relative to biogeochemical inferences made from physical water mass properties alone. Affirmative answers to both these questions would be critical for BGC Argo to have a large impact on the effectiveness of these applications. Moving to the conclusions, I argued, I hope convincingly, that environmentally informed decisions across management time horizons can help us to better meet fisheries, conservation, recreation, and tourism goals. I presented a number of examples highlighting the potential roles of BGC Argo in helping us to attain this goal. I've summarized some of the key points from these examples here. Finally, I raised a number of limitations that could hinder the application of BGC Argo in the fisheries context. While these limitations are non-trivial, I remain optimistic that the novel three-dimensional perspective on ocean habitat that BGC Argo can provide can play an important role in forwarding efforts to improve our living marine resource management and improve outcomes across the fisheries, conservation, and recreation spectrum. I look forward to further discussion of this topic during the panel discussion, and I'd like to thank you very much for your time and attention.